In 1971, I stumbled upon something unexpected while studying a perplexing plant disease. This disease, called potato spindle tuber disease, was causing significant losses in potato crops. Initially, I suspected a virus was the culprit, as viruses were known to cause various plant diseases. However, my research led me down a different path, revealing a previously unknown infectious agent, the viroid. My team and I used powerful electron microscopes to examine infected potato plants. We expected to find viral particles, but to our astonishment, we found none. Instead, we discovered something far smaller and simpler, a tiny, naked RNA molecule without any protein coat. This was a groundbreaking revelation, challenging the existing paradigms of plant pathology. This discovery marked the beginning of viroid research, a field that has since garnered significant attention from the scientific community. It opened up a new dimension in our understanding of plant diseases and the incredibly diverse world of infectious agents. My initial findings sparked a wave of research leading to the identification of many other viroids, each with its unique characteristics and host range. The discovery of viroids revolutionized our understanding of plant diseases and highlighted the importance of exploring the unseen world of submicroscopic pathogens. Viroids are the smallest known infectious agents that cause diseases in plants. They are much smaller than viruses and possess a remarkably simple structure. Unlike viruses, which have a protective protein coat called a capsid, viroids consist only of a single-stranded RNA molecule. This RNA molecule is incredibly small, typically containing only a few hundred nucleotides, the building blocks of RNA. The RNA molecule of a viroid is circular, forming a closed loop. This circular structure provides stability to the viroid, enabling it to survive and replicate within plant cells. The nucleotide sequence of the viroid RNA determines its infectivity and the symptoms it induces in the host plant. Despite their small size and simple structure, viroids are highly efficient at replicating themselves within plant cells. They achieve this by hijacking the plant's cellular machinery, forcing the plant to produce more viroids instead of its essential molecules. This parasitic relationship ultimately leads to the development of disease symptoms in the infected plant. The discovery of viroids revealed a new class of infectious agents that challenged the traditional definition of a pathogen. Their simple structure and unique mode of action continue to fascinate scientists, offering valuable insights into the fundamental processes of life and disease. Viroids, despite their simple structure, are masters of stealth, infiltrating plants through wounds or natural openings. They cannot penetrate the tough outer layer of plant cells on their own and rely on unsuspecting assistance. Once inside a plant cell, viroids waste no time in exploiting their host's machinery for their benefit. Think of a viroid as a cunning imposter sneaking into a factory. This factory, the plant cell, is equipped with complex machinery designed to produce essential components for the plant's survival. However, the viroid, with its deceptive blueprint, disrupts the factory's normal operations. The viroid RNA, carrying its own set of instructions, tricks the plant's machinery into producing more viroids. It's like slipping a counterfeit blueprint into the production line, leading to the factory churning out useless copies instead of the intended products. This disruption of the plant cell's normal functions ultimately manifests as disease symptoms. The infected plant might exhibit stunted growth, distorted leaves, or other abnormalities, depending on the type of viroid and the plant species. The viroids, replicating relentlessly, can spread to other parts of the plant or even to neighboring plants, perpetuating the cycle of infection. Both viroids and viruses are submicroscopic infectious agents that cause diseases in various organisms, including plants. However, despite sharing the same battleground, they are vastly different in their structure and mode of action. Imagine them as two distinct warriors with contrasting fighting styles, both aiming to conquer the same fortress, the host cell. The most striking difference lies in their weaponry. Viruses arrive at the battlefield armed with a protein coat, a formidable shield known as a capsid, encasing their genetic material, which can be either DNA or RNA. This capsid provides protection and plays a crucial role in attaching to and entering host cells. Viroids, on the other hand, are minimalist warriors. They boldly enter the battlefield, stripped bare, armed only with their single-stranded circular RNA molecule. They lack a protective coat, relying solely on their small size and robust RNA structure for survival and infectivity. Their battle strategies also differ significantly. Viruses, with their genetic arsenal, often reprogram the host cell's machinery to produce viral proteins. These viral proteins then assemble into new viral particles, effectively hijacking the host cell to produce an army of viral clones. Viroids, however, employ a more direct approach. Their RNA, acting as a rogue blueprint, directly interacts with the host cell's machinery, tricking it into replicating the viroid RNA instead of its molecules. Section 5. Rogues Gallery: Examples of Viroids and Their Targets. The world of viroids, though hidden from plain sight, is surprisingly diverse. Each viroid species, with its unique RNA sequence, has its preferred targets, specific plant species it infects. Let's meet some of these notorious members of the viroid rogues gallery. Potato spindle tuber viroid, or PSTVD. As its name suggests, PSTVD is a potato plant's worst nightmare. It causes the potato spindle tuber disease, leading to elongated, distorted tubers that resemble spindles, hence the name. This disease can significantly reduce potato yield and quality, impacting farmers and consumers alike. Next, we have citrus ixocortis viroid, or CEVD. CEVD targets citrus trees, causing a range of symptoms like stunting, bark scaling, and reduced fruit production. It's a significant concern for the citrus industry worldwide, as infected trees yield fewer and smaller fruits. Lastly, let's talk about coconut Cadang-Cadang viroid, or CCCVD. CCCVD is the culprit behind Cadang-Cadang disease, a lethal disease of coconut palms. This devastating disease has ravaged coconut plantations in the Philippines and other Pacific regions, causing significant economic losses and impacting the livelihoods of coconut farmers. Section 6. Unraveling the Enigma: Studying Viroids. Studying viroids, these incredibly small and simple infectious agents, presents unique challenges for scientists. It's like trying to decipher a cryptic message written in a language with a limited alphabet. However, researchers have developed ingenious tools and techniques to unravel the mysteries surrounding these enigmatic pathogens. One of the key challenges in viroid research is their minuscule size. Visualizing viroids requires powerful electron microscopes, which can magnify objects thousands of times smaller than the width of a human hair. These microscopes allow scientists to observe the shape and structure of viroids, providing valuable insights into their physical characteristics. Another challenge lies in isolating and purifying viroids from infected plant tissues. Viroids exist in tiny amounts within plant cells, making their extraction a delicate and meticulous process. Scientists use various biochemical techniques to separate viroids from the complex mixture of molecules present in plant cells, obtaining pure viroid samples for further analysis. Once isolated, the viroid RNA becomes the focus of intense scrutiny. Scientists use molecular biology techniques such as RNA sequencing to determine the precise order of nucleotides in the viroid RNA. This genetic blueprint holds the key to understanding the viroid's infectivity, replication mechanisms, and the symptoms it induces in its host plants. Section 7. Combating the invisible enemy: Managing viroid infections. Controlling viroid infections in plants is crucial for ensuring agricultural productivity and preventing economic losses. However, unlike bacterial or fungal diseases, which can often be treated with specific chemicals, viroid diseases pose a unique challenge. There are no commercially available viricides that can effectively eliminate viroids from infected plants. Therefore, managing viroid infections requires a multifaceted approach that focuses on prevention and mitigation strategies. One of the primary strategies for preventing the spread of viroids is using certified disease-free planting material. Propagating plants from healthy mother plants that have been tested and confirmed to be free of viroids can significantly reduce the risk of introducing viroids into new fields or orchards. Another crucial aspect is implementing strict sanitation practices during agricultural operations. Viroids can spread through contaminated tools, equipment, and even workers' hands. Therefore, disinfecting tools and equipment regularly, washing hands thoroughly before and after handling plants, and avoiding the movement of infected plant material can help prevent the mechanical transmission of viroids. Section 8. The importance of understanding viroids. Understanding viroids is not just an academic pursuit, it has direct implications for global food security and economic stability. As agricultural practices intensify and global trade increases, the risk of spreading viroid diseases also amplifies. Therefore, investing in viroid research is crucial for developing effective control strategies and safeguarding our food supply. Viroids can cause significant losses in crop yield and quality, affecting the livelihoods of farmers and the availability of essential food crops. For example, the potato spindle tuber viroid can reduce potato yields by up to 50%, impacting the income of potato farmers and the supply of this staple food crop. Moreover, viroid infections can lead to trade restrictions and economic losses for affected countries. Many countries have strict quarantine regulations in place to prevent the introduction of viroids and other plant pathogens. Outbreaks of viroid diseases can result in trade embargoes, affecting exports and impacting the economies of countries reliant on agricultural exports. Section 9. The future of viroid research. The field of viroid research continues to evolve as scientists delve deeper into the intricate world of these fascinating pathogens. The future of viroid research holds immense promise for developing innovative strategies to combat viroid diseases and protect our agricultural systems. One promising avenue of research focuses on understanding the molecular mechanisms underlying viroid-plant interactions. By unraveling the complex interplay between viroid RNA and plant cellular machinery, scientists aim to identify potential targets for developing antiviral therapies. Another exciting area of research is exploring the potential use of RNA interference or RNAi technology to control viroid infections. RNAi is a naturally occurring process in plants that can silence specific genes. Scientists are investigating ways to harness this process to develop RNA-based therapies that can target and degrade viroid RNA, effectively inhibiting their replication and spread within plants. Furthermore, researchers are exploring the possibility of developing resistant crop varieties through genetic engineering. By introducing genes that confer resistance to specific viroids, scientists aim to create crops that are less susceptible to viroid infections, reducing the reliance on traditional control methods and minimizing the risk of crop losses.